Hi, I'm Saujanya from CSIR IGIB. Neurogenesis or neuronal differentiation is the process where neural stem cells become neurons. This transformation is controlled by various transcription factors that regulate specific sets of genes. Imagine different category of genes as different colored serial lights with the transcription factors acting as switches to turn on specific colors. Our study focused on a telomere protecting protein and its potential as a transcription factor. I'll tell you why in few minutes. Let's take a closer look at telomeres. DNA typically adopts a double helix structure. In the 1980s, it was discovered that the G-rich sequences of the telomeres can form secondary structures called G-quadruplexes. My PI, Dr. Shantanu Chaudhary, was a pioneer to show that G-force occur outside the telomeres and are enriched at the gene promoters. Interestingly, gene ontology analysis reveal that neurogenesis-related genes have significantly more G-force in their promoters. Let's think of these neurogenesis genes as blue lights. Similarly, TRF2, a telomere-protecting protein, is also found outside telomeres and function as a transcription factor by binding to the promoter G-force. With this information, we wanted to explore whether TRF2 could regulate neuronal differentiation genes. In other words, if TRF2 could act as a switch for these blue lights. In neural cells, TRF2 binds to the promoters of neuronal differentiation genes. When TRF2 is lost, it results in the activation of these genes and promotes a neuronal differentiation phenotype. This indicates that TRF2 functions as a repressor. To function as a repressor, it requires partners. Epigenetic complexes such as REST and PRC2 can be thought of as pens that deposit punctuation marks on these genes. These punctuation changes can turn genes on or off. Here, H3K27ME3 mark written by PRC2 is similar to a full stop that stops the gene expression. We notice that with loss of TRF2, the occupancy of these epigenetic complex and the repressive mark reduced on the promoters, indicating that TRF2 is required for the loading of the epigenetic complex to the promoters of the differentiation genes. So the overall summary is that in order to maintain the stem state, TRF2 has to bind to the promoters, recruit REST, PRC2, and deposit H3K27ME3 for the repression of neuronal differentiation genes. And loss of TRF2 leads to activation of these genes, giving rise to neurons. Thus, TRF2 is indispensable to maintain neural stemness.